Well, good afternoon. My name is Joanela Morales, and I am a scientific curator at the European Bioinformatics Institute, and I work on the Locus Reference Genomic, or LRG, project. Today, I will provide a brief overview of the Locus Reference Genomic, or LRG, resource. This is a manually curated reference resource for the reporting of clinically relevant variants. I have organized my presentation in 4 sections. First, I will discuss why do we need LRGs. Then I will talk about what is included in each LRG record. I will then discuss how are LRGs created, and I will end my presentation with how to use LRGs. So first, why do we need LRGs? Well, really, the LRG project came as a result of a meeting that was held back in 2008 by stakeholders who were trying to address a problem. They observed that as our understanding about human genetic variation and its role in disease increases, the accurate description of these variants becomes imperative. However, keeping track of these variants over time is a challenging endeavor due to ongoing changes to the reference assembly and increased characterization of gene and transcript architecture. In addition, the existence of numerous reference sequences and legacy reporting systems make the reporting process cumbersome. For example, consider variant RS72651646 in gene COL-A1A. This variant was reported back in 2008 at the same time that this meeting of stakeholders was taking place. This variant is pathogenic. Here you can see Ensemble's summary of this particular variant. It is a missense variant, and it has a clinical significance. It is pathogenic, as can be seen here. It also is associated with osteogenesis imperfecta. Now, at the time that this variant was reported, the reference assembly was GRCH36. However, as you probably know, the assembly has now changed twice. First to GRCH37 and then to 38. As a result of these upgrades, the genomic coordinates for this particular variant have changed twice. Here you can see the coordinates in GRCH37, and you can see that the coordinates have changed when you move to GRCH38. So the problem becomes, how do we keep track of the precise location of this or any other variant when the reference assembly continues to change? And more importantly, how do we ensure that the community can test and annotate variants with accuracy? This is the particular problem that the group of stakeholders was trying to resolve. And they came up with a solution. They felt that what was needed was the creation of a new system that would address the shortcomings of existing reference systems. This new system, most importantly, would be stable and would be specifically designed for the accurate and unambiguous reporting of variants and those with clinical implications. This new system was called the Locus Reference Genomic, or LRG, project. Now, there are many reference sequences that any user can use when reporting variants. Why should anybody use LRGs? What is unique about LRGs? Well, the most important feature of LRGs I have already hinted at. These sequences, the reference sequences included in each LRG, are stable. They will not change over time. Also, because the LRG project came as a result of a meeting of stakeholders, they were internationally recognized. Also, LRGs are said to be a one-stop shop for each LRG region, and that is because each LRG record includes reference sequences, either genomic DNA, transcripts, or proteins, so the user can determine whether they want to report their variant using genomic, transcript, or protein coordinates. For example, let's go back to our variant in COL-A1A, RS72651646. As I've already mentioned, the coordinates in 37 and 38 have changed. The stable feature about LRGs is that now you can report the same variant using LRG1, which is the LRG for col1A1, and you can use it reporting in genomic coordinates, right here, in transcript coordinates, and in protein coordinates. And as I've said, these coordinates will never change. They are stable, will not change over time. There are other features about LRGs that make them unique and very useful to the community. For example, none of the identifiers in the record will change. There is no versioning needed because none of the sequences will change. Therefore, there's no LRG version 1, LRG version 2, etc., because none of the reference sequences will change. Also, all LRG records are created by consensus in collaboration with locus-specific experts. This means that each LRG has the most suitable sequences for reporting variants at that particular locus. Also, all LRG records are manually curated. Each record undergoes an extensive curation process. And again, the purpose is to ensure that the reference sequences included in each record are the most suitable for reporting variants at that particular locus. Each LRG is also tailor-made for the reporting needs in that particular region. That means that if you look across several LRGs, you will find that none of them are the same. They're not the same to each other, and that is because each LRG is made for the reporting needs in that particular locus. Each LRG also contains the most up-to-date biological information for that region. That means that you can use your LRG to report variants, but also to understand the biology of the particular region of interest. And finally, LRGs are recommended by the key organizations that work on variant reporting, such as the Human Genome Variation Society and the European Molecular Genetics Quality Network. Moving on to the next section, what is included in an LRG record? The LRG is organized in 3 different sections. The first section, the fixed section, as its name implies, is the section that will not change. It's stable. The content of it will never change, and it includes all the reference sequences for reporting. It includes genomic DNA, it includes transcript or transcripts in case when more than one is needed, and the corresponding protein or proteins. The second section, the updatable section, again, its name implies that this section is regularly updated, and the purpose is to include the most up-to-date biological knowledge from both NCBI and Ensemble. Finally, there's the community section, and this section includes information provided by the collaborators about that particular LRG region. Now let's look in detail at each section of the LRG. The fixed section, as a reminder, again, this is the section that will not change over time. It contains all the identifiers. There's the unique LRG identifier. In this case, this is LRG1, the LRG for col1A1. You can also see the HGNC identifier and HGNC symbol for this gene. Requester information is also included in the fixed section. In this case, this LRG was requested by Professor Raymond Dalglish at the University of Leicester. And finally, you can see the sequences, the coordinates, and the identifiers for the reference sequences. In this case, you can see the genomic sequence. You can see the diagnostically relevant transcripts for this LRG, and you can see the corresponding proteins. Finally, there is an exon numbering system that is LRG specific, and it is based on the transcripts that are included in this section. You can see them by clicking on the plus sign next to the exons. If you do so, this will expand and you'll be able to see the LRG specific exon numbering for those who prefer to report their variants using exons. Here you can see the start and the end for each exon within the LRG genomic, the start and the end of each exon within the transcript, the coding sequence, the start and end of the coding sequence, and the start and end of the protein. The updatable section, again, is the section that is updated regularly so that we have the most up-to-date biology on the region. The first component of the updatable section is the mappings to the assembly. We have a commitment to provide mappings to the current assembly and to the previous one. So in this case, you will see the mappings to GRC-H38 and GRC-H37. If there are any mismatches between the LRG and the reference assembly, that will be pointed out right here. You will see a mismatch. And if you use the variant effect predictor, or the VEP, you can then determine the consequence of such a mismatch. If you click on the VEP icon, this will expand and you will be able to see additional information about the variant with its consequence. In this case, it's a missense variant. It is interesting to note that this particular mismatch to the assembly disappears as we move from GRC-H37 to GRC-H38, and now the LRG record is perfectly aligned to the current genome assembly. The updatable section also contains annotations from both the NCBI and Ensemble. Here you can see all transcripts in the LRG. In this case, here are the NCBI transcripts. You can see all the proteins in the LRG gene. Here is the protein for col1A1, and you can see if there are any overlapping genes. You can also see the same information from Ensemble. Here are all the gene annotations for col1A1. Here are the transcripts within that particular gene and the corresponding proteins and overlapping genes. And all this information, meaning the NCBI annotations and the Ensemble annotations together, can be seen in the LRG page in Ensemble. So each LRG, in this case col1A1, has a particular page in Ensemble. If you click on this link, you will be able to see this page. For example, here you can see the page for LRG1. It has the transcript information, the protein information, and then you can visually see, here's the transcript 1 for LRG. Here are the NCBI transcripts, and down here are the Ensemble transcripts. And finally, the community section. As I've mentioned, the community section is meant to provide information that is actually provided by the requester or any collaborator about the particular LRG region. In this case, the requester wanted us to know that there is legacy exon numbering that might differ from the LRG-specific exon numbering. And here you can see, here's the LRG-specific exon numbering, and here is the legacy exon numbering provided to us by the Osteogenesis Imperfecta variant database. Finally, if you're interested in additional data sources for this particular gene, you can click on this link and you will see a list of all the locus-specific databases for this gene. And of course, if you go to each database, you will see all the known variants for that fall in col1A1. The third section of the presentation: how are LRGs created? Well, LRGs are created by consensus with the community. First, an LRG is requested by a requester, and the requester can be an LSDB curator, diagnostic and research laboratories, or a clinician. Once an LRG is requested, a pending record is created. This record undergoes curation. First, experts are contacted for input. The purpose is to make sure that we have included the most suitable reference sequences for reporting variants at that given locus. Experts can help us answer that question. In addition, we, the curators, review all the known NCBI and ensemble data on that particular gene. Once there is consensus on which reference sequences should be included, the pending record undergoes a final check, and then the LRG is published. Once that happens, the requester and all the collaborators who worked with us on the particular LRG are contacted so that they can start using the LRG for reporting their variants. Now, the most important component of each LRG are, of course, the reference sequences. And how are these chosen? Well, as I've mentioned, they are chosen in collaboration with experts. I've already indicated that these experts can be research or diagnostic laboratories, locus-specific database curators, mutation consortia, or even clinicians. The genomic reference sequence is usually the RefSeq gene for the LRG gene. Now, the LRG project is a collaboration between the European Bioinformatics, where I work, and the National Center for Biotechnology Information, or the NCBI. Since the LRG project is a collaboration between the two institutions, it was decided and agreed that the genomic sequence for each LRG would be the RefSeq gene for that gene. And the RefSeq gene, of course, is a reference sequence produced by the NCBI. If there is no RefSeq gene for the particular gene, RefSeq curators at the NCBI will create one. Once the genomic sequence is in place, then we need to decide which transcripts to include as reference sequences. As I've indicated, the transcripts can be suggested by the requesters, but they undergo review by the LRG curators. Now, the manual curation of transcripts is extensive. We use an in-house alignment pipeline for this. All known transcripts, for example, in this case, all the ensemble and the NCBI transcripts, are all aligned to the assembly. Then evidence for each particular transcript is assessed, and finally, cross-references are checked. In this case, for example, we have checked the consensus coding sequence for each transcript. We have checked the UCSC transcript level support, where 1 is given to transcripts that have the most evidence and 5 for those that have the least evidence. We can also see here if their annotation is manual or automated. Only transcripts that are diagnostically relevant are included, and only transcripts that are approved by both the curators and the requester are included. In this case, you will see that it was decided that this particular NM 000131.4, would be the transcript for this LRG for F7. You can clearly see that this transcript is the longest one, and it also contains all the exons that have supporting evidence. In terms of LRGs that are available for the community to use, there are over 900 LRGs in the pipeline. Of these, 655 are available for the community to use immediately for reporting variants. Our LRGs vary. We have LRGs for cancer predisposition genes, for example, BRCA1, BRCA2, TP53, genes in the Fanconi anemia pathway. There are also LRGs for inherited cardiac diseases, and these LRGs, I should say, were created in collaboration with the British Heart Foundation. There are also blood-related LRGs for blood-related disorder genes, and these were created with the university, in collaboration with the University of Cambridge. And we also have genes that have been included in the UK NIQUA's quality schemes for clinical laboratory testing. The final section of this presentation will cover how to use LRGs. Well, the purpose of the LRG is to describe and report variants using LRG coordinates. So the first use of LRGs is to describe your variant using either genomic, transcript, or protein reference sequences, or all of them. In this case, this particular variant has been reported using genomic coordinates, transcript coordinates, and protein coordinates. Now, it is very important to make sure that once your variant has been described in using LRG coordinates, that they do follow the HGVS conventions for variant reporting. For this, we recommend that users use the Mutilizer software, which will tell you if you have made any errors when describing your variants using your LRG record. We also believe it is very important for variants to be submitted to the public domain, in this case, dbSNP and ClinVar. And we have created a process whereby users can submit their variants into these two databases through the LRG. And this can be found at our website at lrg-sequence.org, submit variants. You can also use the LRG record to view and interpret all the variants that sit within your LRG of interest. You can see the specific variant that you're interested in, or you can look for all the variants that fall within the LRG region. And if you're interested, you can also look at the consequences for each variant using the variant effect predictor, which I mentioned earlier. If you're interested in seeing a specific variant within your LRG, this can be done by going to the ensemble section within the updatable section of your record. And if you go to the link that says See Variants in Ensemble for this gene, and you click on that, it will take you to this page within Ensemble, which shows you the LRG transcript for this gene. It will then show you all the NCBI transcripts for reference. You can see the consensus coding sequence, this one here in green, and at the top you can see all the ensemble transcripts within your LRG. At the bottom here, in this square, is your variant of interest. And as you can clearly see, this variant sits within an exon. The most important thing to note is that the coordinates for this variant using LRGs will not change. However, if the same variant is reported using any of the other transcripts that you can see here, those coordinates will change. The only coordinates that will be stable are the ones in LRG. If you're interested in seeing all the known variants within your LRG region, again, you can do that by going to the ensemble section within your record. And again, you click See Variants in Ensemble for this LRG. This will take you to the LRG page, and you will see the variation table. And this table provides a list of all the known variants within the record, and you can see their coordinates in LRGs. And you can see additional information about the variants and their pathogenicity. If you're interested in all the known phenotypes associated with the LRG region, you can also see that in Ensemble. Again, go to the Ensemble section, but this time click on the link that says See the phenotype diseases associated with this LRG. That will take you to a similar page, but it will show you all the phenotypes. Here you can see all the phenotypes that are associated with this LRG region for col1A1. And now I would like to give a live demonstration, a brief overview of the LRG website. For that, you can use any browser. In this case, I'm using Chrome, and it's lrg-sequence.org. You can find your LRG of interest if you know it. Typing LRG1 in this case, it will take you to the LRG page for the LRG of interest, but you could also type your gene of interest, and you should get the same exact result. If you then click on the LRG identifier, that will take you to the LRG page. This should be familiar as I've shown you screenshots, but again, here are the identifiers, LRG1 for the gene of interest, the HGNC symbol and identifiers. Here are the requester information, and here is the fix section. You can click on plus signs to see as much as you want or as little as you want. In this case, if you could click here, you will see the genomic sequence of the LRG in Small letters are the introns, big letters are the exons. Here you can see the start codon highlighted in yellow, and you should be able to see your stop codon highlighted in orange. Here's a transcript information. In this case, there's only one transcript, NM000088.3. And here is the sequence. Now in this case, in blue and black, it's hard to see, but you can see the exon boundaries with the start codon, and in small letters you can see your untranslated regions. Again, the stop codon in orange. Your exons, again, the LRG specific exon numbering. In this case, this transcript is quite long. It has 51 exons. You can see all the start and end sites in the genomic DNA, your transcript, your coding sequence, and your protein. Here is a translated sequence for this particular transcript, and this is the protein reference that will be used for reporting your variants. Going down to the updatable annotation, you can see the mappings to GRCH38 with no mismatches, your mapping to GRCH37 that has the one mismatch we've already discussed, and your variant effect predictor link. You can see here the NCBI annotations. If you click plus, you can see the gene annotations, the transcripts, the proteins. You can hide that and only see your ensemble section. Again, gene annotations, transcripts, proteins. And here's the community section where you can see your exon numbering, the legacy exon numbering, which I've mentioned the community provided for us. Here's the LRG specific numbering that goes from 1 to 51. Here is the legacy numbering, which for the first half of the transcript, they are the same. However, the second half, the exon numbering changes. And the purpose of this is so that you can clearly see that any variants reported using the legacy numbering system in exon 35 now correspond to exon 34 in the LRG specific exon numbering system. You can also have alternative amino acid numbering, which function in the same way as I have already described. Going back to the ensemble section, these are the links that allow you to see the LRG page. You can see the variants in ensemble, and you can see the phenotypes and diseases associated with the LRG region. If we're interested in seeing the same variant that we discussed before, you can click on that link. It will take you to the ensemble page, the LRG page within ensemble. Here's a variation table, and you will be able to see all the known variants in the LRG gene. Now we know that our variant of interest is a missense mutation, so we can filter by looking for missense variant. If you do that, you'll be able to see your filtered table, and here is our RSID that we've discussed previously. Again, here's the RSID, here's the coordinates in the LRG, and you can see information is clinical significance, and the fact that it is a variant, as a missense variant, and additional information in terms of pathogenicity. Again, you can click on it if you're interested in more about this particular variant. And this is the view that I showed you in my presentation. But below here within Ensemble, you can find additional information on the genes, the genomic context of this variant, population genetics for this variant, phenotype data, and all sorts of information about this variant of interest. Again, if we go back, here you can see this should take you again to the LRG page, but in this case you'll see the phenotypes associated. And you can see that this gene is associated with several different kinds of diseases, and here's our osteogenesis imperfecta that I've mentioned is associated with that particular variant we've discussed. And this concludes my presentation. I hope this information has been helpful. If you have any questions or you would like to contact us, there are several ways of doing this. I have included a couple of ways of, if you want to request or submit or ask for any help with any of the LRGs, you can contact us at any of these email addresses. But if you have any question about this presentation or any LRG, feel free to contact me at jmorales@ebi.ac.uk. If you want to read more, you can read our most recent paper. And if you want to look through any of the frequently asked questions, you can do so at our website, lrg-sequence.org. Before I finish, I'd like to acknowledge my colleagues here at the EBI, at the NCBI, outside collaborators who have contributed significantly to this project, all the collaborators who have worked with us on all the records that we have public, and then our funding sources. Again, here's our website, here's our paper and papers for you to review.